So they basically gave me 2 words to tell me what I should do, and they were wet, which I guess means not dry, I don't know, in BASIC. So I filed a Title IX complaint, and it emerged that they actually meant, biology by wet. So that's what I'm supposed to do. Again, as Clay just said, there's sort of parallel tracks going on. One is this one, which is sort of basic background on how RL has been used in brain and behavior. And I think they have in mind kind of an intro for people more from the CS side, or maybe kind of neuroscientist psychologists coming from outside the area. It's going to be pretty basic. Michael Littman in the other room is doing an introduction to computational RL. He's amazing. He's wins a lot of teaching awards, which I don't actually. I'm married to the general chair, which I think is probably how I got the job. So I recommend Michael Littman. But you found this room, so here we go. So I'm going to try to make this kind of an overview. So I'm going to stay at a pretty high level. So I'm going to kind of imagine that you have a sort of ML machine learning kind of perspective on things, but I'm going to be very sloppy about notation and so on. I hope you'll be able to see the machine learning poking through, those of you who are familiar with it. I'm not spending a lot of time explaining machine learning ideas. Along the way, I'm going to try to sort of introduce kind of brain ideas and brain methodologies. And kind of my goal, in a way, is to give you kind of a feel for the range of data that have sort of been brought in contact with reinforcement learning ideas, sort of what we know and how we know it in a way, and how we think about it from a reinforcement learning perspective. So to give you a smattering of examples meant to convey that. Okay, so I think reinforcement learning is a nice story because it sort of shows 2 ways that neuroscience has really been helped by computer science, which are linked but different. And I'm going to focus mostly on the first day. So one is conceptual. So one of the things about neuroscience is that it's often hard, what the brain does is so complicated that it's hard to study it scientifically or quantitatively. So if I'm studying vision, and I have a neuron that I think is involved in color vision or motion vision or something, it's sort of obvious what you need to do to test that hypothesis, right? I move things around or I change their color and I see how the neuron fires. If I'm studying learning or decision-making or reward or value, it's a lot harder to know, to quantify what it is I'm trying to study. And so many of us have looked to computer science both for a kind of definition of what it is that these things mean, like to make a decision or to make an optimal decision or to optimize value. And also, to go further than that, to kind of give us a kind of process level algorithmic explanation for how, as a sort of hypothesis for how the brain might approach these problems, right? So that sort of goes beyond, economics, I think, has typically focused on defining what the optimal answer to a decision problem would be, but hasn't thought a lot about how you might arrive at something, given that those are often intractable, that might approximate that. So the nice thing about I think reinforcement learning in particular is it provides a very nice bridge from kind of step-by-step manipulation of data back up to what it's trying to accomplish, right? And a sort of mapping between those 2 things. And the other thing that's kind of related is sort of analytical. So we can think of machine learning algorithms or reinforcement learning algorithms as basically likelihood functions, right? As generative models of where our data might have come from that are testable kind of in terms of raw data, whether that's neural spikes or functional brain imaging or choices made by people or animals. And so we can sort of view analyzing data as itself kind of machine learning problem of making inference from noisy measurements with a generative model. So that'll be a little bit in the background along the way, but that's sort of a big idea in my area. I should say, so this is a tutorial and I don't really have a particular agenda. So interrupt me if you have questions and so on. Let's definitely not let me talk for 3 hours uninterrupted. Okay, another thing I'm trying not to accomplish here is sort of the opposite thing, which is sort of using neuroscience to inform AI. Like I'm a unabashed consumer of reinforcement learning. I read about it and try to understand how it might help me do my job. There are a lot of people who are very interested in the idea that we're going to sort of reverse engineer how the brain works and use that to improve AI. And that might indeed happen, and there are little hints of that indeed in the reinforcement learning literature, which I think in many ways was launched when Rich started looking to psychology for inspiration about AI. But I think what I'm going to talk about today is mostly situations in which we've learned from them rather than vice versa. And just to make that point a little bit clearer, another thing we're not engaged in is sort of working from, trying to figure out how the brain works de novo. Like, we're bringing ideas from reinforcement learning to try to make sense of data. So this is a famous figure of the connections between areas in the monkey visual system, and each box is a region of visual cortex or subcortex, and all these lines are projections, and it's always just a mess, right? Like, and this is just at the level of areas. If you think about the level of 100 trillion neurons or something, there's no way starting with the raw data is going to get you anywhere. You need some idea of what it's trying to accomplish. This is a figure of the number of pages per year, per edition, plotted against year in the primary graduate textbook in neuroscience. Kendall, Schwartz, Jessel, and gets more and more authors every time too. And the point is, it's a log scale, so it's growing exponentially. This is actually Daniel Wolpert's figure. And it's horrific, right? You project this to the future, and my children are not going to be able to carry their book to school. And so the point is that neuroscience is producing a lot of information, and the goal of theory of us is to bring sense to that, to bring understanding to it, and the book is to make this book shorter. And right now we're kind of losing, but there's still hope. Okay, so that's where we're coming from. So then I'm going to talk about really two issues today. One is a sort of classic story that now goes back about 20 years about how the brain may implement a sort of prediction error-driven temporal difference learning and the involvement of the neuromodulator dopamine and its targets in that learning. Talk about behavior and neurorecordings linked to that, and how that has been recently extended to the cases of more interest in reinforcement learning, which is sequential decision tasks. And then I want to go somewhat sort of beyond that idea towards the idea that there are multiple decision systems in the brain, that this is just one of them, the different ways of the brain's implementing sort of sequential planning, and various other extensions. Just again, going behind this core kind of classic idea, just to give you kind of, again, an overview of the state of the field. Okay, so just getting started. Of course, computer scientists are well familiar with the bounded problem. You choose between a set of slot machines or options, you get an outcome, and you repeat that, you try to figure out, you try to maximize reward. We do this with people and animals all the time. This is a really kind of a workhorse task for the study of learning and decision making in biology. And roughly guided by computer science, the kind of strategy that practitioners in biology have in mind when they're sort of studying this task is sort of 3 steps. It involves imagining that people or animals are maintaining some kind of summary variable associated with each slot machine, like its expected value, expected reward minus cost or something. So we shock people for choosing these, which is good fun. And then they're going to choose, maybe the best, maybe with some exploration in there, and then they learn, they get some outcome, and then they learn and update their predictions. This is sort of the basic story, and one we'll come back to over and over again. So this is sort of a core task. I'd actually like to start with a simpler task, which actually gets rid of the choice and just focuses on the prediction and learning, and that's Pavlovian conditioning, right? So now going back more than 100 years to the work of Pavlov, Pavlov argued that you could study basically prediction and how predictions are learned from experience from behavior directly. And the idea was if you pair, as you probably know, a neutral stimulus like a bell, initially neutral stimulus, with some biologically important outcome, like in his case, meat powder. I'm not sure what that is, but it was what he used. If you feed the animal meat powder and ring the bell in anticipation of the meat powder over and over again, then what you find, of course, is that famously the animal starts to salivate and also to produce digestive juices, which were Pavlov's real interest, in anticipation of the meat powder. So you ring the bell, and before you go in the meat powder, you see this behavioral response. And the idea is this provides it's a sort of quasi-automatic or reflexive. It provides a kind of behavioral window on the animal's expectations about reinforcement. And it's importantly, it's sort of outside the context of a choice problem, right? It's just he's just standing there and you're putting stuff in his mouth and you're reading about, off directly from his behavior, how his beliefs develop. So it's meant to be a kind of pure study of prediction without other things like choice or decisions getting in the way. And this has more recently become of interest again because people have sort of new, it's a well-developed set of behaviors and people have new neural and mathematical and computational tools and so on that they want to apply to it. And so this sort of stuff has come back off the shelf and gotten to be of interest again in the last couple decades. So the classic theory in this area, which you probably know, is basically the delta rule, right? So Rescorla and Wagner in 1972 suggest the way we can think about this is the animal needs to predict at a sort of a trial level the amount of food that he expects as a function of whatever stimuli he sees in that trial. So say a bell or a light, these are sort of binary indicator variables, and they each have a weight associated with them. So the idea is that the animal sums up the weights associated with the things that he sees in a trial, and that controls how much he slobbers or how much reward he expects. And then the learning rule is supposed to be just error-driven. You compute the difference between the food you got and the food you expected, R minus V, and you update whatever stimuli were present to reduce that prediction error with a sort of step size alpha. So machine learning people will recognize this as, you know, a special case or an instance of a thing that has been used all over the place, like the Kalman filter and many other areas. One thing I just want to mention about this is you can also just by rearranging this update rule, if we reduce ourselves to the case of one stimulus where I show you a light and I give you meat powder over and over again, then all this is really doing is estimating the expected reward in a trial. This weight, if you just rearrange this algebraically, is really computing an exponentially weighted running average of the rewards previously received during that stimulus. This delta rule is just a rule for calculating the weighted sum of what you're predicting before and the rewards you got now. If you express that in terms of the rewards itself, so you could think about the weight that it's predicting, the amount of food it's expecting is just the exponentially weighted sum of the reward you got last trial, the reward you got 2 trials ago, and 3 trials ago, and 4 trials ago, and so on. So it's got this, it's just exponentially windowed kind of running average rule, and that's all that this prediction error does. And I mentioned this because we're going to try to get this shape back out of behavior a few times, and so I'm going to come back to that. But that's the sort of the theory that arose in this area a long time ago. Just to give you a kind of feel for the kind of data that Rescorla and Wagner used to argue for this, they're phenomena like blocking. So again, you've probably hopefully heard of this. You don't need to just pair lights with food. You can do sort of multi-phase things where you ask, if I pair a light with food, a bell with food, if I pair a bell and a light with food, does what I learn about the light depend on what I know about the bell? Okay, and the prediction is what we know is that if you pair the steak with the bell in this first phase, the animal learns to salivate to the bell, right? And the question is, if you now go on to do the second phase, where you pair the steak with the bell and the light, what does it learn about the light? And the answer, according to Rescorla and Wagner and according to data, is that learning about the light is attenuated. That is, the slobbering, if you present the light alone, is less than it would have been if the animal hadn't already predicted food in the presence of the bell. Yes? So the idea is you're controlling for, it's not mere salience either. So the idea is that, by the way, you should probably ask questions into the microphones because there's video going on, I think. The video guy gave me a thumbs up, so there you go. So let's see. So salience. So let's say we just omitted this first phase, then the relative salience of the light and the bell would determine how they sort of split out learning to predict this steak, right? But if we train the bell up to asymptote, the idea is the sort of prediction error driven learning idea is that in the second phase, there's no prediction error, there's nothing left to learn. So the bell already predicts it, whether or not it was more or less salient. There's no prediction error, there's no learning, and there's no nothing left to attribute to the light. And of course, the data aren't perfect. It's not like there's actually no learning to the light, and it's not, I'm sure salience matters. But this has been taken as evidence for this kind of error-driven learning dynamics. Right? This is the classic phenomenon behaviorally. Okay, just one more example of a phenomenon from this area that's not well explained by the Rescorla-Wagner theory, and which we'll come back to once we have temporal difference learning in hand, is called second-order conditioning. So now, and we'll see variants of this kind of task a number of times over the course of today. You pair the bell with the stake again, but now I pair the light with the bell without the stake in phase 2, right? And so we have a situation in which the bell is a predictor of food. Let's say we don't do this so many times that the bell loses its prediction. And the light has never been paired with food at all. In fact, it's been paired with the absence of food that you expected. So on the view of Rescorla and Wagner, this protocol should actually make the light, if anything, predict kind of negative food, right? Predict the absence of food you should have gotten. Its weight should be negative. You should get negative prediction errors whenever you see the light, and because you see the bell and you don't get the food you wanted. But in fact, at least depending on the particular parameters of the experiment and so on, you can get positive responding to the light. Again, although it's never been paired with food to the animal at all, as though it sort of inherited it from the bell. It's called second-order conditioning, and that's not something that Rescorla-Wagner explains. Now this is an important point because particularly when we get back to the choice domain, this is thought to be a sort of laboratory model of the fact that most of the reinforcers that we actually kind of work for in the real world aren't the kind of biologically significant ones that are inbuilt. So there's sort of, water and food and sex and shelter or something that are supposed to be like the basic, biological building blocks. But I usually say when I'm teaching that, I'm here teaching for money, although in this case I'm not being paid, so I'm not sure why I'm here, but in my typical lecture, I'm teaching for money, and money is of course nothing but pieces of paper, but pieces of paper that I can use to purchase food and water and so on, that is neutral stimuli that have been paired with biologically significant events. And this is supposed to be kind of a laboratory model of how things like money achieve the status of rewards or something like it. And again, we'll come back to this, and this should be familiar from a reinforcement learning perspective. Rescorla-Wagner doesn't get it. Okay, so let's talk about the neural basis for all this. So that this was sort of where things stood in the 70s. So let's start talking about the brain. So this is a sort of cut through the human brain down the middle. The spinal cord comes up here into the brain stem and the midbrain. Outside is the cortex. And there's a particular group of neurons that are of particular interest in this area. They're in a couple of small nuclei, sort of clusters of cells. The cell bodies are clusters of cells down here in the midbrain, but they send these very diffuse projections. So neurons have axons and dendrites. Their axons go up to contact other neurons sort of throughout many of the interesting parts of the rest of the brain, sort of the forebrain, frontal cortex, and the striatum. So these ascending projections, this small group of neurons have these very broad projections, and they release a particular chemical, a neuromodulator called dopamine. So as you also may know, neurons, communicate by spiking, right? So it's it's a hybrid electrical kind of chemical code. So a neuron spikes, an electrical impulse travels down its axon, and it releases, at its synapse where it meets other neurons, it releases some chemical that crosses the synaptic junction, impinges on the other neuron, and does something such as produce electrical activity on the other side. Dopamine is one of those chemicals. There's many others. So these neurons use dopamine as their neurotransmitter, and they're very localized. There are very few of them in this part of the brain. So it was known for a really long time that these were sort of interesting, and really in two areas. So there were a group of people interested in them because they were interested in reward. So these neurons seem to be involved in, they're the target of essentially all addictive drugs in some way. They sort of mimic or replace dopamine or release dopamine. These self-stimulation experiments where you electrically stimulate some neurons and let animals lever press to do that, and if you do it with dopamine neurons, they'll basically starve to death lever pressing because they like it so much. At the same time, they seem to be involved in motor control. Yeah, Rich.
I know you talked about how the synapses, or the neurons, the axons go out and they connect to other synapses. But is that true of dopamine, or does dopamine just act more generally on the tissue?
Yeah, so I think the answer is both, actually. Dopamine is interesting because when it's released, we'll see in just a second, the neurons fire several spikes very rapidly, and that produces a big release. So much so that it overflows the synapse. So it is released at a synapse, and there are receptors at the synapse on the receptor neurons that are waiting for it and hear it, as it were. It also has a more volumetric signaling property where it overflows the kind of local synaptic reuptake mechanisms, gets out in the extracellular fluid and diffuses. And there are also dopamine receptors not on synapses that pick up this extracellular dopamine extrasynaptically. They have a higher affinity, they're more sensitive, and some of them are on dopamine itself, so dopamine neurons are self-regulate by doing this, but other neurons monitor this as well. So that might be, I think that's relatively unusual. It's probably not the only example of it, but it's true that that's one thing that dopamine does. Yeah, so, and I have something more to say about that later, actually. The other thing these neurons are involved with, which isn't obviously the same thing, or really obviously related, they also seem to be involved in movement. So there was another community that was sort of studying the same system because these neurons are, for instance, relatively selectively killed by Parkinson's disease. So Parkinson's disease is a degenerative neurological disorder in which more or less these dopamine-containing neurons of the midbrain gradually and selectively die, de-innervating their targets across the forebrain from their dopamine input. And if you've ever met a Parkinson's patient, sort of the primary kind of complaint they go to the doctor with isn't anything to do with reward or addiction or anything. It's sort of paralysis, right? So they have difficulty moving, slow movement, tremor. And so there was a neurological community interested in movement and the biological basis of movement and movement initiation and so on. That was also kind of interested in these neurons. And so these are two separate kind of worlds. And then Wolfram Schultz in the sort of late 80s started recording from these neurons. And he was kind of from the movement camp, I think. And he was sort of surprised to see they didn't have, for the most part, responses that were linked to the animal's movement. So he published a lot of sort of confusing papers for a while about what these neurons were doing, which didn't meet his expectations. And sort of they're summarized in this famous figure. The semantics of these figures, so neurons, again, remember they signal by releasing electric spikes. So this is a single dopamine neuron, but it's typical of the ensemble. Each line is sort of a trial, and it's aligned on some event. Time is on the x-axis. It's aligned on some event in time. In this case, is the animal receiving a drop of juice in his mouth. So the animal's a fluid-deprived monkey. This is an electrical recording of the neuron spiking. It spikes. These neurons spike at a background rate, which is low. This is a histogram across trials of the firing rate. And what you see is when you drip juice in the thirsty monkey's mouth, the neuron releases a burst of 3, 4, 5 spikes very rapidly, right? Just what I mentioned. And then it goes back to this kind of slow background firing rate. And what was separately reported in a series of papers is that it's not as simple as these neurons just care about reward. So for instance, if you do a sort of Pavlovian conditioning experiment and you signal the reward on the basis of a preceding stimulus like a fractal on the screen, then the response transfers. They don't respond to the drop of juice anymore. They respond to the stimulus. And again, it's a very well-timed kind of phasic burst response. And indeed, if you tell the animal he's going to get juice and then you fool him, you don't give it to him, what you see is this kind of well-timed, it's sort of hard to see often because the background firing rate is low, but notwithstanding that, you see this kind of well-timed pause in the firing of these neurons around the time or right after he should have expected the reward. So basically two different groups from the reinforcement learning community around the same time saw these papers and kind of put two and two together and said this is like a prediction error, right? So the response to the stimuli, we're going to come back to, we're going to focus on the response to the reward or the non-reward for the moment, although the stimuli is really where the action is. So just to make this a little more quantitative, this is another recording from Chris Fiorillo. There are now 5 different fractals here that signal reward with different probabilities. So this is a fractal that signals reward always, and this is a fractal that signals reward never. And in between is in between. So there's a 50-50 one and so on. And what this is the response to reward following the 50% Q or the 75% Q or the 100% Q. And what you see is, this is basically what I showed you before. If you're not expecting reward, you get a big response. For something for sure, you get no response. And in between, there's a graded degradation. And conversely, this positive response to this transfer to the Q is a graded thing. If I'm expecting reward 50-50, I get a 50 response here and a 50% response here, right? So, as I said, computational groups kind of seized on this and realized this is like a prediction error. So, again, just thinking in terms of this Rescorla-Wagner model, we expect some particular reward on the trial, and we view the response of the neuron as being a sort of baseline level for 0 plus the reward you expect, the reward you got minus the reward you expected. Then you get a positive response when you're not expecting a reward, nothing when you get the reward you expected, and a negative response signaled by an inhibition, presumably, when you don't get the reward you expected, right? Yes?
So you had the picture that was showing that, or like the no reward following predictive cue. Is there anything? Are there any papers that kind of talk about over time, if you repeatedly train them to expect it, and then you stop giving it to them, but you repeatedly keep giving them the cue, the change in those levels slowly, I'm assuming slowly going back to the normal behavioral.
Yeah. So there is that's called extinction, and there is work on that. It's a bit of a long story. There's work on dopamine neurons doing that, and they do kind of what you'd expect, although they have kind of weird triphasic dynamics. So it's almost like there's positive and negative responses that are kind of engulfing each other to kind of cancel each other out in the end. So it's not as simple as you might have hoped, I think. But actually, the behavioral account of extinction itself from the Rescorla-Wagner model is not as simple as you would have hoped either, because there's all kinds of evidence that extinction is not actually well explained as you train away the prediction, but rather you're learning something new that kind of overrides that prediction. And that's a much longer story, which again I think I'll touch on a little bit later, but that might be what you're seeing here, that you see some combination in extinction of new learning and old learning that are sort of superimposed. You actually see sort of complicated dynamics within this little pause. That's going way beyond, that's my interpretation. But all this, I should say, is kind of not during learning. This is kind of the steady state, right? This is the animal, has been trained for months on these images. And even so, he gets prediction errors. When you see a cue that you only know 50% whether you're going to get reward or not, you have a prediction error every time, right? You can't know for sure. So this is a sort of well-learned kind of condition. Okay, so good. We can be a little more quantitative about this. Hannah Bayer and Paul Glimsher had a really nice series of studies, or one really nice study, in which they had a monkey do a very complicated task, the details of which don't matter for what they ended up doing with it. The animal got different amounts of juice on each trial, and sometimes 0, which he couldn't really predict. And so you can think of it as being a kind of situation in which he's just continually trying to maintain a prediction of how much reward to expect, and he's continually producing a prediction error. And so she then recorded from dopamine neurons and did a sort of regression analysis. So when she tried to express the response to the dopamine neurons, the number of spikes, as, well, let's look at this one first. So she figures out how much prediction error she thinks the monkey is experiencing in each trial, the difference in what he expected and what he got. And what you see is a rather linear relationship, right? So when the prediction error is high and positive, the dopamine neurons fire a lot. As it gets lower and lower, the response falls off until it's below the background firing rate. And then it looks like it's sort of rectified because you can't fire below 0 spikes, right? So this, at least within the positive and weakly negative regime, you had this very nice quantitative relationship between prediction error and the neural response. Another way to say that is you can do a regression analysis in which you try to express the response of the neurons now as a weighted sum of the rewards you're currently getting and the rewards on each preceding trial. 1 trial ago, 2 trials ago, 3 trials ago, 4 trials ago, and so on. And use regression to estimate the best weights to explain the neural response on the basis of the rewards the animal got and the numbers of spikes its dopamine neurons gave you, without making any assumptions about the form other than linearity of this function. Right? So you do linear regression, and what you pull out is that the neuron is positively modulated by the reward it's currently getting. So it's like R minus V. So this is the R. And then it's negatively modulated by all the rewards that came previously, so the minus V. So if on the rewards I got previously, those taught me how much reward to expect. The most recent one being most strongly weighted, the next most recent one being next most strongly weighted, and so on. So this negative part, in a sense, represents a weighted sum over the previous rewards, except it's negative because it's subtracted from the response because it's R minus V. She's helpfully turned it upside down here so that you can see that if you focus on this and then look at it from right side up, it looks like this, right? It looks a lot like an exponentially weighted average over the past rewards, which is what the Rescorla-Wagner kind of error-driven learning model predicts this function should look like, right? Yes, Michael.
Is the sum of those negative coefficients on the lags, is it about the same as the coefficient?
That is a good question. It looks to me like it might be a little off. In another data set I have, it's definitely off. I can tell you that from my own data. I forget whether Hannah checked that, but you're right. The prediction of the model should be that if everything's really working out, this sum should equal that sum, right? It doesn't look like it does, and it definitely doesn't in my data. So there may be imperfections. In a sense, it may be overweighting the current reward. It may be additional activity related to the current reward, or it may not be completely canceling out the reward. There may be a tendency towards the positivity. That also might relate to this rectification, right, in some way. So there's this nonlinearity here that's a little bit difficult for this analysis. But you're absolutely right. In the data set I've been looking at just recently, that's definitely the case. It's overweighted. Good. But notwithstanding that, this I think is a pretty nice match between theory and data. This is not now. It's not a learning curve. It's sort of a function that tells you how the firing rate of the neuron depends on recent history, and it's the function that's implied, more or less by the theory. So this is, I think, a pretty nice success. There's also been work trying to sort of test this idea causally. So just recently, for those of you who aren't in neuroscience, there's been a sort of advent of a revolution of kind of biological tools that allow neuroscientists to control the firing of particular subgroups of neurons that are well biologically identified genetically, directly in time. And so I already mentioned that, addictive drugs mimic dopamine, which is sort of consistent with this story. These electrical stimulation experiments are consistent with the story, but it's very hard to know for sure from those experiments that what's really doing that what's really having the effect is specifically the stimulation of dopamine neurons at particular times and so on. So recently, using what's called optogenetics techniques, researchers have been able to directly activate identify dopamine neurons with particular patterns and test whether they have effects on things like classical conditioning, pavlovian conditioning. So trying to think which example to go through. The data is a little dense. So they've done it with blocking and it works. Let's do extinction. So this is the thing one of you asked about. So if you, this is behavior. These are 3 groups of animals, 2 control groups and the optogenetic group. If you train the animal, he will produce responses that are like the animal slobbering. These are mice, and so they spend time in a port rather than dogs slobbering. This is the behavioral response, suggesting they're predicting something. If you then keep giving the cue and stop giving the sucrose, that response dwindles away. But if you pair that extinction experience with stimulation of dopamine neurons, you attenuate that effect. So it's as though you're blocking or overriding the negative prediction error that was driving this effect, and you attenuate this extinction, right? So they're able with timed activation of dopamine neurons, time to the extinction event, they can block this extinction, and they see a difference between here and here, between the animals who got the stimulation and these various control groups who didn't. So this is good causal evidence that these neurons are actually kind of playing the role that they're supposed to. Okay, and that brings us basically back to choice, right? So that, again, all this was experiments without decision making in it, and so of sort of limited interest, I think, to reinforcement learning people, arguably. But again, we think of that as being an important kind of subroutine in decision making, right? So we think about our sort of back of the envelope model of a bandit task is you're learning values tied to each of the slot machines by basically this same error-driven learning process, and that is that's driving your choice, right? So it's like the same kind of learning process with a different kind of behavioral expression. So we and others in animals have worked, we and humans and others in animals have worked a lot with this kind of task. Just to show you the particular kinds of data sets I'm going to show you, we've looked at ones in which we often use non-stationary rewards. So this is one in which this is called, you might know this is a restless bandit. So the reward probability and the shock, we shock people also, the shock probability associated with a particular slot machine is taking a sort of Gaussian random walk over time. So this one starts out bad, a high probability of shock and a low probability of reward, and ends up kind of more middling. And this one starts out good and ends up sort of still good, well, bad for a while. So this allows us to sort of study, not to test whether people find the best bandit, like if the blue one just pays off money, do they find it and then just keep choosing it? That's sort of not of particular interest to us because we pay for them to make choices while they're in the scanner, so we want to keep them learning kind of in the steady state. So we want to sort of watch them chase the rewards rather than watch them approach asymptote. The kind of behavior we see in a task like this, so this is a test just with money. So what's on the y-axis is how much money that we paid the subject. On the x-axis is the last 100 trials of a choice test. This is one subject's data, and she's choosing the green slot machine over and over again, and its value drops a bit, and she hunts around, sees if she can find something else. That doesn't work. She finds yellow, finally sticks with that for a while, checks out green again, and so on, and eventually switches. So this is kind of the raw behavioral data, and you can see, in effect, kind of learning and exploration here. One way to kind of interrogate this, again, kind of going back to test the error-driven learning idea, we'd like to characterize the function that relates kind of the recent history of outcomes to the choice the subject's going to make on trial T. So I make a choice here. I'd like to understand the function that takes my recent history of rewards and shocks and choices to that. One way to do that, again, is basically with linear regression, or in this case, logistic or multinomial logistic regression, since the outcome variable is a multinomial rather than a spike count or something. So what's plotted here is a regression coefficient that says if I was rewarded for choosing the slot machine one trial ago, say the red slot machine, then I'm 4 logits more likely to choose it again. So I'm much more likely to choose it again if I was rewarded for it. And if I was rewarded for choosing a slot machine 2 trials ago, or 3 trials ago, or 4 trials ago, or 5 trials ago, then again, I'm more likely to choose it again. So there's this sort of familiar form to the function that it starts high and falls off kind of rapidly. Comfortingly, if we shock people, it drives them away from the slot machine for the most part. So here we can see, again, if I shock you for choosing the red one trial ago, you're less likely to choose it on the next trial, and same for 2 and 3 trials ago, and again, that falls off kind of rapidly. Okay, so this is not perfectly well fit by that, again, that kind of exponential decay, but it's close enough that people have taken, and the interesting difference is like the fact that it doesn't seem like it asymptote. There might be a couple different time scales to it or something, so that might be interesting to something we've looked at. But to a first approximation, people have taken this again as kind of this sort of evidence, which is seen very widely in humans and animals, as again behavioral evidence from choices now that the decision variable is also kind of driven by this error-driven learning dynamics. Okay, and indeed, if you fit these data with a two-parameter reinforcement learning model, it does a pretty good job of picking up the choices. In fact, better than this, given all the free parameters. Just as an aside, another way to say this is that I've come pretty close to viewing the kind of temporal difference learning model as a likelihood function or generative model for choices in this kind of a task, right? And we could think about, people have done a lot of sort of fitting these models now where they're not kind of generic regression models, but sort of structured reinforcement learning models to data, and tried to argue about which model, which learning rule or whatever does the best job of explaining sort of trial by trial choices. So this is for a data set like this one, except simulated, the likelihood function implied by a learning rate on the one hand, and a softmax temperature, sort of choice noisiness, which is like the height of these logic of these choice functions on the other. And so this works, so this is the simulated parameters and this is the recovered parameters. But you see these models which are sort of designed for computer science aren't especially identifiable or sort of well-behaved as statistical models of choices. So you see these kind of banana-shaped likelihood functions that couple the two parameters and so on. So this is just sort of a technical point. These models are not especially well-behaved in the psychological and neuroscientific setting, although people use them a lot. So it's challenging at times to sort of fit these models and identify them and assess them because, of course, they weren't really designed for this. The parameters aren't independent, aren't well identifiable, and so on. But that's, again, just a sort of aside.
Hey, Nathaniel.
Yeah.
Sorry, over here.
Ah.
So I'm just wondering, is there a reason that you're using shocks rather than having them lose money? I mean, would you get similar results?
Yeah, that's interesting. That's a long, let's see. So you indeed do get similar results with losing money, and people have done that. Here we use shocks. We're concerned about, sort of framing effects or house money effect. So we wanted something that was, and we've used also kind of bad tasting juice and stuff and good tasting juice for the same reason. So in this experiment, we really wanted something that was frankly aversive. If you come into an experiment, that, we can't actually take money away out of your pocket, right? That you're going to go home with what you started with and no worse. So I can't make you stay and wash dishes for me or something. So we're not sure that taking money away from you, if it's our money anyway, is really rightly thought of as punishing you in quite the same way as just giving you a big old electric shock. So that's the answer, and I think it is in fact the case that there are well-known kinds of framing effects, and in the case of monetary loss, and exactly what controls them, I think is not particularly well understood. And so my read of the literature in this area that uses monetary loss is that it's pretty messy. So both within a study, there's more variation between subjects, maybe in how they construe the outcomes. And also across experiments, lots of things seem to matter. You get reversals in the direction of neural activity and stuff, depending on things we don't even know what they are, right? So things that something to do with the methods or the framing of the task. And so that's why that's why this particular study wasn't done that way. But it's a good question. Obviously, that in itself is an interesting area of study and worth understanding better. Okay. So this is just one example, and perhaps not the most impressive one, but it comes from humans and it uses the same analysis I showed you before, so I thought I'd put it up on the screen. This is from Rob Rutledge in Paul Glimcher's lab, and this is Parkinson's disease patients. So as I said, Parkinson's disease patients actually go to the doctor because they have trouble moving, right? The doctor gives them dopamine replacement therapy and they're okay. But once people started getting interested in these reward learning models of dopamine, they thought, well, these guys must also have cognitive problems, and they're known to have cognitive problems, but they must have problems with reward learning. Specifically, we should be able to see it in their if we have them do bandit tasks or something. So people did that, and they do. So this is, I think, the most important comparison here is probably patients on and off their medication. So you have patients. So it turns out the medication that's used for Parkinson's disease has a very sort of short half-life, and so your life is you take the drug and you're good for a while, and then you sort of sink into paralysis again. You take it again, and so if you give the drug to them if you give the task to them right before versus right after their sort of daily dose, then you give yourself a comparison, and the effect's not incredibly impressive, but again, this is one of these choice functions, and you see it looks less steep off the medication than on. So another way of saying that is once you put them on the medication or the on minus off comparison, the learning rate, the sort of step size, the thing that dopamine is supposed to be doing, driving a change in your reward prediction, gets sharper, right? And that effect is significant. Healthy elderly people are sort of not too different from patients off their medication in some of them, but it's a sort of slightly longer story. So that's an example of causal evidence for dopamine involved in choices, again, according to this reward learning theory. Okay. Let's talk about recordings now of prediction errors and so on in people. So in people, we, for the most part, can't stick wires in their head and record the firing of neurons, but we have other technology that is the best we can do. And so in particular, many of us have worked with a technology called functional MRI. So MRI is magnetic resonance imaging. It's usually used to measure structural properties of the body, but this is functional because what you're measuring is blood oxygenation. So we're trying to measure the usage of the brain in something like real time and not just its structure. So the thing that you're measuring is called the blood oxygenation level dependent or bold signal. And it's based on the fact that when hemoglobin, the molecule that carries oxygen in the blood, is oxygenated versus deoxygenated, it has different magnetic properties, and you can pick that out by this giant superconducting magnet. So the brain is functionally modular, right? So different parts of the brain do different things. I mentioned dopamine neurons doing some things and other places doing other things. And that's presumably because of wiring and so on. It's efficient to keep functions closely together. So that means that we can actually see changes in blood flow locally when people use different cognitive capacities, right? And that's because, moreover, all this electrical activity that carries information from one neuron to the next uses a lot of energy, right? So, in fact, quite a great deal of energy kind of maintaining the voltages across the neuronal membrane that carry these spikes. The brain spends an awful lot of energy pumping ions in and out of the neuron to maintain that and allow spikes to propagate rapidly. Maintain these synaptic potentials or these cellular potentials. And so that uses blood oxygen, energy, which drives blood oxygen, and then that changes the local blood in an area, and you can pick that up with this machine. So this has relatively good spatial and temporal resolution by the standards of these things. So you can pick up, in the standard version, voxels, which are 3 millimeter cubes, and temporal resolution of maybe 5 or 10 seconds, which reflect not the limitations of the machine so much the limitations of the vasculature, the way blood flows in. So the kind of thing you see, so this is an early experiment with someone set in the scanner for just hours and hours and hours, or maybe days, and listen to a word, and this is sort of word-triggered average of the bold signal, the hemodynamic signal in his auditory cortex, and you see this is where the word happened, a reduction of blood oxygenation, and then the prominent thing is this big increase, 5 or 10 for the following 5 or 10 seconds, as blood flows into the area to replace to this sort of overusage or over response that vasculature sort of apparently overcompensates for usage, and you get this positive bold response. It's the same thing with visual cortex in monkeys. You get a similar response. There's a reasonably, across areas, not especially the same, but a reasonably stereotyped response. So again, it's kind of slow and localized and mostly positive and has this sort of event-related dynamics, okay? Again, this is because some of you might do computer science. I'm just mentioning all this. And then you probably see all over the popular press and papers these sort of highlighted brain areas, right? So what's going on here is these highlights represent voxels in which some statistical test came out true. So you run some test over the whole brain, and you say, where was the bold activity higher when I was thinking about cars versus thinking about airplanes or something? And if that is true here, you color that in yellow. That's called a contrast. So the most famous ones are faces and houses, right? So there's particular parts of the sort of visual object processing hierarchy that respond selectively, relatively selectively to scenes versus faces. There's a lot of fighting exactly what it is that actually means. But just to give you an example of what the data looks like, this is a task when people are looking at displays that consist of colors, faces, and houses all the time, but they're paying attention to one or the other and answering a question about it in different blocks. So if you look in the so-called fusiform face area, you see while the people are thinking about faces, paying attention to faces, you see this response, and it's quiet otherwise. If I'd shown you the time course for the parahippocampal place area, you would have seen activation during the time of thinking about houses. Okay, so that's a sort of classic contrast. And a broad set of findings interesting to us is that different sorts of contrasts related to reward or anticipation of reward tend to activate a couple different areas which you might recognize as being the kind of areas that I mentioned are targeted by dopamine. One is the striatum, and the other is, that's shown here in a different projection, the other is the ventromedial prefrontal cortex. So this is a meta-analysis of some 200 studies that was recently done. Just to give you a feel for kind of data that underlie this, here's 6 different studies I picked at random. So this is people expecting more or less money. You see this nice ventromedial prefrontal activation, higher when they're expecting more money in a bandit task, looking at attractive versus unattractive faces, getting a squirt of Coke versus Pepsi if you like Coke more, or Pepsi versus Coke if you like Pepsi more, in the famous paper on culturally familiar drinks. This is striatum now, so ventral striatum for winning or losing money, for smelling a food that you're sick of versus not sick of, or not sick of versus sick of, and for unpredictable versus predictable juice. So this last one is looking more like a prediction error, and we'll come back to that. But the main point of this is that across all these different kinds of reinforcers, primary and secondary, food and money and Pepsi and so on, this sort of common pattern of responding suggests that there's a very generalized appetitive function that involves this circuitry, right? Yeah.
So at least those first pictures looks like it's in the frontal cortex that the big response is. And I guess I thought that people said it would be in like the basal ganglia.
So there are multiple parts of the brain that turn out to care about rewards. We are indeed going to focus more on the basal ganglia going forward, but there's a whole other literature on this area. So there's a lot to say. First of all, these areas are interconnected, right? And they're both targeted by dopamine. So this is medial prefrontal cortex, ventromedial prefrontal cortex, and orbital frontal cortex below it. That's also historically a lot involved in appetitive and rewards and utilities, and neurons there care about different kinds of juice and so on. They project to these same parts of striatum, and they're connected in a sort of loop, which I'll talk about in a bit. This is the striatum. This is the striatum viewed in a different slice, right? Slice this way, and that's the kind of area you're thinking about. Right? But these two areas come up again and again.
Part of the basal ganglia?
The striatum's in the basal ganglia. Yes, I'm sorry. So the striatum is the area that you've probably heard the most about. But it is true that there's this whole other area nearby that's connected with it that also has similar activity. And it's been of interest too. I could have just omitted it, but it's there. These are examples of that, and these are examples of striatum. you sometimes can see the striatum. It's hard to get them both in the same slice, actually, but all these studies basically report both activations. Which is not to say the activity is the same. This looks more like a prediction error, and this looks more like a prediction, basically, as it turns out, or maybe an outcome signal as well.
I just have another quick question.
Yeah.
Sorry, I was wondering if there had been any studies that showed. So you're showing that certain things, houses go in a certain area, and just going back to the other part where you were talking about how like a bell can signal juice. I was wondering if you had looked at any sort of thing where you associate a specific house with juice, and then or with a reward, and then if you show them houses but not necessarily that house, if that's more strongly. If you do see some sort of like dopamine boost, and if depending on the experiment, maybe if you showed them only a house and then gave them a reward, if that increase of dopamine to other houses is stronger than if you show them a specific house and juice and then a specific house and not juice. And sorry.
So there's, so there's two separate things going on in that question. One is people use faces in houses because they're easily decodable, and so there are lots of experiments that use faces in houses. And I'll give some examples of that, maybe not the particular one, because we can then look in the brain and see if people are thinking about them. But there's also work, all over psychology and conditioning on generalization, right? So, whether it's, using bells of different frequencies or indeed using, stimuli that have, subclass relations with different houses or different faces or something. There is work on that. I happen to know Liz Phelps right now is working on a study involving, much like you said, generalization between different instances of a category. It doesn't have to be a face or a house, right? It's a face or house for technological reasons, as it were. And people generalize, and there's typicality effects and stuff. And it's not something I know a huge amount about, but it certainly is that kind of stuff. People have also looked at conditioning in which I train you that a face predicts a house or a light predicts a tone or something. I look for changes in visual versus auditory activity that's evoked by this, or house activity evoked by a face, and so on. Is that? Yeah. There's all this kind of stuff. Yeah.
Sorry, Nathaniel, still on houses and
Sure.
faces. So, is there any thought on how much of the response in all these different areas is based on the semantics of it or the meaning, and how much is just on visual patterns? I mean, we see a house; it's like, okay, right angles, lots of lines. There's some nice straight line segments in it, whereas faces are all curvy and sort of a bit more full of gradients. Is there any thought that it's mainly just essentially the patterns in the data as opposed to what a house or a face actually means? I know that's a tricky question.
Yeah, that's a tricky. I mean, I don't think it's as simple as low-level visual features. You could do controls like scrambled houses and stuff that maintain edges and things, and it's not that. So it's got some semantics to it. And it's not my area, but there's a vast knockdown, drag-out fight for decades about how to interpret this stuff. So is it faces, or things that, faces are things that you're really trained to be expert in classifying, or they're socially important? What is it about faces, right? There's tool selective areas also. What's that really about? I really mentioned it more as an example of something that you can do, of an fMRI response that's clear. And we're going to later on talk about a few examples where we employ it instrumentally as opposed to kind of scientifically as something, by doing things like pairing houses with things and seeing if people are thinking about them. But yeah, it's an interesting area. There's all this work on vision, which I'm not going to really qualified to talk about.
So
Yes.
I'll let you get on in a minute. I just want to ask you something that
Sure.
you mentioned very briefly a second ago, which is you said sometimes the areas light up in response to a reward or a prediction error or the outcome. And these seem to be quite different things. And how do you discriminate? Do you discriminate them in terms of timing, and are they likely to be reflecting activity in different neural systems?
Yeah. So, I mean, there's a lot to say about that. And when I hold that, and I'm going to say a few more things about exactly that. But I should stress, remember, we're talking about the signal here, which is a relatively poor temporal resolution, right? So this is, a response that, not because in the neural responses we've seen can be very fast. This response is 5 or 10 seconds long. So telling apart different events can be tricky. So we're going to rely, to the extent we can, on whether those events are, independent statistically, on whether if I turn the reward amount up versus the prediction up, can I change, which function does it follow? So people have done that pretty carefully and dissociated the three signals you mentioned across these areas, which are of course collinear. So we'll talk about that in a sec. But yeah, so that is important. Again, we're not measuring neurons firing here now. We're measuring blood oxygen metabolism, right? So it's got something to do with neural activity locally, but it's not a slam dunk. It's not easy to interpret. So that said, here's what we've done, or lots of people have done. You can generate a time series. This is sort of a fancy version of faces versus houses. Like, let's imagine that I take every time the person or animal the person gets a reward, I write down how much I think his prediction error was, R minus V, what he expected minus what he got, based on some fit model. I make a time series of that, which has an impulse, like what I think a kind of Wolfram-Schultz dopamine neuron would be doing. Now I'm going to, just like the impulse response functions in the monkey, right, where I flash a checkerboard or I flash a word, I'm going to convolve that time series with a hemodynamic response function. So it's going to be smeared out by the hemodynamic response, and I'm going to get shifted and scaled copies of the hemodynamic response, assuming linearity, which is a pretty good assumption. And then I can just ask where in the brain does the bull signal covary with, say, this prediction error time series? Where does it look like the dopamine signal? So tons of people have done this. John O'Doherty was the first to do it in this exact way, but this has been replicated lots of times. And what you see is these areas that we just talked about lighting up again. And it's hard to get them in one slice, but they're basically the same areas that are innervated by dopamine. Okay, so this is the striatum here, which is usually shown in this slice, coming in the other way. And this is this ventromedial prefrontal area, right? And in the striatum, this prediction error related responding is really strong. It's really, so normally, bold activity is pretty noisy. You got to average a lot to see stuff. You don't usually kind of look at raw data. You usually look at these kind of statistical tests. But if we look at striatum, which is again is part of the basal ganglia, the sort of most prominent target, densest target of dopamine, if what Yelniv did is she did a task in which is sort of modeled after those monkey tasks I showed you before, in which subjects were expecting different amounts of reward and then got different amounts of reward and had sort of different kinds of characteristic prediction errors, like reward predicted and then gotten, partly predicted, then gotten bigger than you thought, or smaller than you thought, or you got exactly nothing and nothing, and so on. Pretty much the same conditions we talked about. And the prediction is, accounting for the hemodynamic smearing, you get dynamics like up and down, or up and over, or whatever. And she just hired an undergraduate to circle the ventral striatum on a bunch of people's brains individually and pull out the signal and average it locked in time, which is actually relatively hard work. So I tried this, and my undergraduate quit after the first day because it's really laborious, but they're better at Princeton. And there's a pretty amazing match, in my view, and again, by the standards of this kind of stuff, between what you expect and what you get. So there's a really strong response in this area. And again, it's not dopamine, right? It's metabolism. But if you look at this response in Parkinson's patients or in people given drugs that strengthen or weaken dopamine, you strengthen or weaken the signal. So there's some indication that what you're seeing is sort of an effective dopamine in this area, okay? So this is sort of as close as we have to a human measurement of the sort that looks like these monkey measurements that we talked about before. So we think of this as being a sort of a manifestation of the same thing. Yeah.
So I've always wondered, maybe you can clear it up a little, where do the dopamine neurons get their input from? That they're so all-knowing that they know all these reward contingencies already.
Well, so actually one answer is the same neurons in striatum. So these neurons are in a sense the strongest inputs and the strongest outputs of the dopamine neurons. But there's tons of other inputs, and people have done a little bit of work kind of working backwards. So there's a very prominent set of work now actually on negative inputs of the dopamine neurons from a set of neurons in places like the lateral habenula. They're supposed to carry about punishments and missed rewards and stuff, and they seem to inhibit dopamine neurons. So Hikosaka has been involved in that.
How fast does a dopamine neuron fire after a reward?
Yeah, so that's another story. So that can be fast or slow depending. You can go back. It can be as fast as like the short latency response. I was going to say, I think it's like 100 milliseconds. Even looks a little faster than that here. It depends a lot on basically, so the really short latency response turns out to come from if you're a neuroscience superior colliculus. So you don't really need a lot of neural processing to figure out like what part of the screen is lit up or something, which is, it turns out, all you need to solve a lot of these tasks. If you give the animal something that takes some time to figure out, like a random dot, motion thing that takes a few 100 milliseconds to resolve itself, then presumably that involves a more cortical pathway, and the dopamine responses in response to that are slower. But there's been a lot of arguments about this. There is a fast kind of non-cortical connection into the dopamine neurons that drives a lot of this, Schultz's original stuff, I think.
Cool, thanks.
Sure. Okay. Okay. Yeah. Actually, so maybe I'll make a few sort of general, just since again I'm assuming some of you are outsiders to this area, and even if those of you are insiders, about fMRI and what it's good for. Just seemed like a useful thing to do. So this is sort of an aside. So one thing to say is so you think about, and what these pictures show is kind of where in the brain, does the bold signal correlate with some model time series, like faces or houses or prediction errors or stuff. And I guess what I generally would say is that, this was very exciting like 20 years ago, but people are not, for the most part, that interested in asking where in the brain does stuff happen. It's sort of not really an answer to a question. You sort of want to know how does the brain work, right? So it was indeed interesting to see, 10 years ago that dopamine target brain areas look like the dopamine signal. But since then, the question we've really been interested in is, now this is like faces and houses, right? We can look in this area and ask, how does prediction error behave? If this is a neuroreported prediction error, what happens if I change your experience in this way or that way? If I do blocking or I do extinction or whatever, what happens to the signal, right? So we're kind of moving into the question, not, where is it, but kind of what, going back to it and asking what it does. And so those are sort of more interesting questions. The other thing is, as we've already discussed, there's lots of different variables that might be correlated, right? So I've written down a prediction error here, and I've said, well, it correlates with these parts of the brain. But of course, the prediction error is partly driven by the outcome reward. It's also partly driven by prediction, so it's correlated with both those things. And that can be very pernicious and hard to tell apart. So I can tell you when we talk more, the striatal prediction error is really well verified, but in general, there's lots of studies where people run a model and come up with some time series and say it's got correlates all over different parts of the brain, and they sort of declare success and like their model is right. But of course, lots of different models will produce that result, and lots of different time series from the same model will produce that result. And so you have to really think hard about kind of confounds. And I think people would kind of be blinded, tend to be blinded by sort of computational modeling to not think about, if I told you that part of the brain responded to houses and I didn't control that properly, I think you'd probably catch on faster. But there's lots of examples where it's hard to know what's really happening in these areas that apparently correlate with decision variables and stuff. So one example is prediction error, right? So I just said prediction error correlates with all these parts of the brain. I talked about these two, but I showed you, I think it's the same picture. This is the cerebellum. This isn't even, the main part of the brain. This is the posterior cingulate cortex. This is a different part of frontal cortex. And so a number of people have looked very carefully at prediction error. And in response to your question, one way to define prediction error is really, it depends on 3 separate time series. So there's prediction error in the general case we haven't really talked about yet at the time of the stimulus, right? That the dopamine neurons respond to the stimulus, and then they show this difference between the reward, the outcome time that they got, and the one that they predicted. And so one way to kind of break this down is do all 3 of those tests separately. And if you look across all these different areas, which kind of roughly correspond to some of these areas and more, the only one that actually passes all these tests and really is kind of a prediction error that is being the sum of these 3 independent pieces is striatum, and all these other areas respond to one or two of the subcomponents, right? So for instance, this ventromedial prefrontal area responds predictively at the time of a stimulus, and it responds to the reward itself at the time of an outcome, and that's why there's not a star there, but it doesn't actually, it's not a prediction error, it doesn't respond, that response to the outcome is not modulated by the prediction, but it's good enough that it lights up, okay? So that's a cautionary note, but also, interesting, right? Yes.
Yeah, so all of the prediction errors we're talking about so far are reward prediction errors, is that right? So, I mean, something, of course, quite a few of us in the room look at as prediction errors of other things that might not be reward as well. Can you comment on this, on the prediction error localization for non-reward kinds of predictions? Is that outside of scope for today?
Yeah, I had a slide about it way at the end. I can say a few things about it. It's kind of a mess. So I mentioned already. So let's just take the case of punishment, right? I mentioned already, if you just take money away from somebody, it seems like sometimes you get a prediction error that's consistent with this one. That is, if things are worse than expected, you get a suppression of the bold response and so on. Sometimes you get the opposite. It's in the same place often, but it's upside down. So I showed you behavioral data from a task we did with shocks, and I think that one, it's oriented as you'd expect. It's money and shocks kind of live in the same boxes on the same axis, but there are other ones where it seems the other way. So I think some of this is framing, and some of this might be that they're actually, again, we're not really specifically recording dopamine here. This is people, and there's lots of inputs to these areas, and there's other neurons doing other things. The kind of embarrassing thing is in monkeys also, there's a lot of confusion about this. And this has led to some fighting about these theories as to whether these neurons are doing something more attentional and not something reward related. So I think there are indeed some kind of dopamine neurons in some places that look like a prediction error and kind of reward minus punishment, and some that look like they just code prediction error, violations of expectations more generally, so they respond positively to unexpected shocks and stuff, and I'll show some data about that later. I think the best guess now is these are anatomically separated, they project different places or something, but it's surprisingly hard to get this straight. I think we know less about the psychology of learning in the aversive case too, so I gave the example of expectations of punishment and so on, but that's only one of the problems. More generally, even in rats trying to lever press for safety or something, it's thought they learn this by framing safety as a positive goal and stuff, and so there's sort of there's a bunch of stuff going on, and you can't really hurt monkeys in the lab for ethical reasons, and so they don't really punish them as much as you'd really like them to. For if what you wanted was to understand this better. So you get these kind of half-ass experiments where they threaten to, squirt them with a mildly annoying air puff to the eye and they don't actually do it and stuff. And so it's like, what do you make of that, right? So that's the situation. It's a bit of a mess, but I'll come back to that again. Yeah. Yeah. Yeah, that's a fair point. Cerebellum is involved in various kinds of conditioning and prediction and so on too, and there's other kinds of prediction of other stuff and other kinds of surprises as well, and the brain's clearly doing that. We'll talk more about that too.
So sorry to dwell on this point, but suppose you do experiments where you have a moving object that goes behind a screen. There's an expectation that it's going to come out, but it doesn't, right? So this is the kind of experiment where you have an expectation and might be violated. There's going to be an error that gets produced. Have people looked at where in the brain this might live? Does that have any similar?
So we've
mechanism?
looked. I don't have a slide of this. We looked at basically an MDP. So it's not quite. It's more stylized. But we looked at kind of state. I don't know if Aaron Bornstein was in here somewhere. We've done a couple of experiments. We've looked at expectation violations in a Markov decision process. Jan Gloscher is on one of them. Aaron Bornstein's for southern one of them. I think the main answer is the kinds of areas that you do see graded expectation and expectation violation signals in these cases. That you get a state transition you don't expect, or don't go somewhere you did expect. They're not really, if they're in striatum at all, they're not in the same place in striatum. But a lot of what you see is sort of attentional circuitry and parietal cortex, cortical circuitry that's arguably involved in attention is the most prominent response. And again, that might be useful instrumentally, whatever it's really doing. You might be interested in reading off expectation violations from those systems, right? Okay.
Bottom line there. I mean, it could be that we don't know. We don't have as good a neuroscience handle on where these other prediction errors are happening.
So I think the answer is the core story about dopamine and reward and reinforcement learning seems like it's got a lot going for it, and like it's, there's something right about it. It's going to be more complicated and whatever, but I think it's basically what I'm telling you is basically well accepted. I think how it extends into the punishment domain and how punishment learning works and avoidance learning, whether it involves the same system, and if so, under what circumstances and so on, is much less well understood.
But we're trying to ask the question, what if it's not reward, not punishment? It's just other things that happen that you might predict.
So to some extent, that's also I think not as well understood, but it's also kind of traditionally the domain of kind of perceptual neuroscience, and the whole cortex is sort of doing that, right? It's sort of making predictions. The sort of Bayesian generative view of vision, right, is this kind of interplay between, or efficient coding or something, is making predictions about sensations and updating when they're not right. So this is sort of thought to be kind of outside our domain. There's lots of work on it, and I don't think, I don't mean to declare it outside the scope, but it's sort of more, it's more than just this. It's sort of part of a different, it's part of perception, right? Where that's where it's been worked on more. You don't look happy with that.
So I think you're saying it hasn't been thought of this way.
It hasn't, and we've thought of it in terms of model-based reinforcement learning. So where do you get world models, and how do you make predictions about state transition MDP or something? But most of the, once you get into more generally into violations of sensory expectations and formation of sensory expectations and stuff, there's just a huge body of work on that under the domain of perception that I'm not sure I'm going to be able to completely unpack with precision. But it intersects importantly, and state construction obviously is important as well, which is in some sense the same thing as perception, arguably. So, and again, we'll come back to this a bit, but it, we've sort of started in the middle with reward, and we're going to start to work out to things like state expectations and what is state is a sort of newer question that's still ongoing. Okay. Okay, how are we doing on time? Let me do one more little block of stuff, and then we can take a break. I wanted to talk a little bit, just putting the dopamine system, situating a little bit more in terms of what it's connected with. Again, just to give you a feel for the kind of story that people have in mind, and the kind of data that that people have thought about in this context. So this is mainly concerned with what are the sort of targets of dopamine. What's if this is driving learning and decision making, how's that happening? And finally we come to the basal ganglia. So I mentioned striatum a few times. What are the basal ganglia? So this is a slice through the brain this way. It's the same one I've been showing you. The cortex is the sort of sheet of cell bodies, right, that they're sort of wrapped around the brain and very wrinkly in people's to give it more surface area. But it's kind of a flat sheet, right, with a few layers. And then underneath it is mostly wiring, right, these white matter as axons that carry signals from these cortical neurons, to and from the spinal cord, right? Floating in that white matter are these sort of lumps of cell bodies which have weird kind of uncomfortable shapes that are hard to keep track of, and have terrible Latin names that are really hard to keep track of. And they're collectively called the basal ganglia. They're ganglia because they're chunks of neurons, and basal because they're in the sort of base of the brain. Yeah, so that was what, here, see striatum. So the other thing is, it's just such a mess. These areas were named by anatomists in the 19th century in Latin before the kind of wiring was understood. So like the striatum happens to be cut in half. Like this chunk and this chunk are, really should be thought of as being part of the same nucleus. But there's a big white matter tract, at least not in rats, but in people and monkeys, that cuts right through it. And so it got two different names, which we only later renamed as being collectively striatum. Right? And that's all over this area. There's two things that have the same name because they're next to each other, but they're different in terms of circuitry, or they're two things that have different names. And it's not my goal at all to school you in this stuff. Like, if you want to learn that, you should think twice. But anyway, don't ask me. It's just a mess. But the point is, so yeah, striatum can be also referred to as caudate, putamen, and nucleus accumbens, which are three different pieces of it. And it has this incredibly weird shape, right? So this is this thing, the caudate nucleus, wraps around here, has a tail, and the piece we've been looking at is up here at the front. The putamen is also coming off of it. That's striatum, right? And that's the one we're mostly talking about. But just to get a little bit more feel for what people think they're doing, again, I mentioned this has really come up a lot. These areas, the basal ganglia, are mostly the domain of kind of people who study movement. And the other thing people thought about the basal ganglia a lot, kind of textbook-wise, prior to all the reward and decision-making business, and it's, this is, I think, a little bit important, is that they're somehow involved in producing and withholding movement. So the thing labeled putamen here, which is the striatum, it's in all these other areas with acronyms, which I'm not going to unpack, are other basal ganglia nuclei, and this is cortex. And it turns out the cortex has a kind of reciprocal relationship which is topographic with different parts of cortex. So cortex and striatum, I should have said, cortex projects kind of topographically to different parts of striatum, and then that projection is kind of reciprocated through a loop through several other areas, and in fact 2 loops, arguably, and it's all very messy, but there's a sort of functional unit here, which is this loop from cortex to striatum and back to cortex again for different parts, and they're different loops you could think of corresponding to different areas of cortex and different areas of striatum. And the reason I'm mentioning all this is because it was noted in the kind of medical literature on these disorders of movement that one of these pathways has 3 negative signs in it, and one of the pathways has 2 negative signs in it. So the ones that are colored black inhibit neurons, and the ones that are colored white excite neurons. And so you have inhibit, inhibit, excite, inhibit, or inhibit, And so if you make it through the whole loop, you end up either having a net excitatory and inhibitory effect on these, say, motor cortex neurons that are thought to control muscles. And it was argued that this is sort of like a kind of, they're called the indirect and direct pathways, like a gas and a brakes on behavior, that they can sort of initiate or produce muscle movements, or they can hold them back, depending on how many negative signs you've got. So you have a facilitatory or a inhibitory effect. And the imbalance between these two pathways gives rise to all these diseases like Parkinson's, where you have impoverished movement if you have too much inhibition, like in Parkinson's, or things like tics or other sorts of diseases like Huntington's disease, where you have too much inappropriate movements, these sort of structure, these chorias, they call them, these weird movements that Huntington's patients have, arise from the imbalance between these two pathways. And this is an incredibly influential idea, so much so that, there's sort of a backlash against it, but there's something sort of right about it. And whenever I go to the doctor, I say I'm a neuroscientist, they say, oh my God, I hate the basal ganglia, I had to learn about that in medical school, and I'm like, let's focus on my appendix, please. But this is a really big deal in sort of medical neuroscience. And the point is, this is the striatum, this is the circuitry that dopamine is projecting into, right? So you think of this is the sort of the thing that's stopping and starting movement, and then you've got dopamine neurons projecting right on here and controlling that. That's the sort of connection that we think of as sort of probably driving this kind of reinforcement learning function, right? That's the sort of idea. How much more about that do I want to say? I'm going to say a couple things. The principal neuron type in striatum is called the medium spiny neuron because they have these spines, and this projection from cortex into striatum sort of lands on the if you do a sort of electron microscope graph, which is this cartoon of, lands on the end of these spines, and that's where that synapse is that we talked about, dopamine crossing the junction into the neuron. But dopamine neurons come and touch the same spines, and they make their synapses too. So cortex projects striatum and into these loops that control behavior. And dopamine's right there, but it's like on the end of the spine, on the sort of root of the spine instead of the tip of it, as though it's like getting in between, like physically, to take control of the signal, right? It's like it's right in the place to kind of be the gatekeeper between cortex into movement. So, and you have dopamine here. So we think of this as this synapse or group of synapses from cortex into striatum as being a candidate for at least one of the places where dopamine might affect the strength of connections with cortex and striatum that affects movement, that sort of drives reinforcement learning. That's the kind of big picture idea. It's indeed the case that dopamine affects the strength of synapses. I won't unpack that further. And so altogether, just sort of back up, and then maybe we could take a coffee break. This story ranges from, effects of individual synapses to behavior like animal salivating, and people have been arguing that conditioning effects suggest error-driven learning, that these dopamine neurons seem to carry this prediction error for reward, and dopamine neurons follow this pattern. They may train predictions in striatum at the synapse where they affect plasticity. So there's this story that spans synaptic events all the way to behavior, and it's a little bit of a cartoon, but it seems like there's something right about it as well. So that's sort of what I want to set up, and what we'll talk about in the second half is sort of somewhat more modern stuff that sort of builds on this foundation.